साइंस प्लॉट प्रो स्मार्ट एनालिसिस बेहतर पब्लिकेशन आज ही आजमाएं. वी ऑल नो टी टेस्ट ANOVA correlation and regression. The real issue is different. Science has changed, data has changed, journals have changed, but in many cases, our statistical thinking has not changed at the same speed. Today, our data are no longer simple. We collect repeated observations, multiple treatments, time points, biological markers, environmental variables, and interacting factors. But very often, we still reduce all this complexity into 2 bars and 1 p-value. This is the gap I want to discuss today. The gap between the complexity of modern science and the simplicity of our usual statistical practice. Many times statistics becomes a ritual. We collect data, run a test, search for PA less than 0.05, and then decide whether the result is important or not. But science cannot be reduced to this mechanical act. A significant p-value does not automatically mean biological importance, and a non-significant p-value does not always mean absence of effect. This problem is not limited to one discipline. In medicine, confounding is often ignored. In ecology, complex environmental relationships are simplified too much. In psychology, continuous human behavior is sometimes forced into artificial categories. In agriculture and biotechnology, repeated growth or response data are often analyzed using endpoint t-tests. So the issue is not lack of data, the issue is how we reason with data. Earlier, we often asked only one question: does it differ, yes or no? But modern research asks deeper questions. How strong is the effect? Under what conditions does it occur? Does it change over time? Through which mechanism does it act? How probable is the conclusion? What is the uncertainty? These are more mature scientific questions, and they require more mature statistical tools. High-impact journals increasingly expect more than a p-value. They want effect size, confidence intervals, proper model selection, diagnostics, reproducibility, and clear interpretation. In other words, journals are asking us not only to report significance, but to quantify uncertainty and justify the method. This is perhaps the central message of the introduction. Statistics is not simply mathematics applied to data. Statistics is scientific reasoning under uncertainty. Whenever we analyze data, we are not only calculating, we are making a judgment. We are deciding what model fits the question, what assumptions are reasonable, what uncertainty remains, and how cautiously the result should be interpreted. In future, the strongest researcher will not necessarily be the one who generates the largest amount of data. Many instruments can generate data. The stronger researcher will be the one who can interpret complexity correctly. This is where statistical thinking becomes part of scientific maturity. So in the next parts, we will move through 4 modern ways of thinking. First, longitudinal models, when data change over time. Second, probability-based modeling, when outcomes are not simply continuous. Third, causal balancing, when groups are not naturally comparable. Fourth, Bayesian reasoning, when evidence is treated as a degree of belief, not just a yes or no decision. The aim is not to make statistics difficult. The aim is to make analysis more faithful to real science.